Hello my dear students, welcome you all. Again, let us continue the class on biomolecule. Okay, this is our fourth lecture on biomolecules. So in the last session, we have discussed about the structure of protein. Means we just have started what to discuss in the today's class. Okay, here when we say the structure in terms of inorganic chemistry, structure means molecular formula like KMnO4, CuSO4, something like. When it comes to organic chemistry, it is the study of a two-dimensional structure. When it comes to physical chemistry, it means for a three-dimensional structure. But since we are studying this concept in biology, we have to study the biomolecules, mainly the proteins, in terms of a primary structure, secondary structure, tertiary structure and a quaternary structure. Okay, as we all know the process how a protein is formed, by the arrangement of amino acids or binding of many amino acids with the help of a peptide bond. Okay, we should know how these multiple structures are formed, how these are named. Okay, that's about our today's class. Right, we'll go to the topic, that is structure of proteins. We'll discuss about all these 3-4 concepts. It means the primary structure of protein, the secondary structure of protein, tertiary structure of protein, and lastly we will discuss about a quaternary structure of our protein. Okay, today's class will be very short since we need to finish up the proteins, and in the next class we will go to the next biomolecule. Right, so coming to the class, structure of proteins. As we all know, proteins are the heteropolymers. Made of strings of amino acids. Strings are a chain. Strings or chains of amino acids. For example, 1 amino acid, 2 amino acid, 3 amino acid, 4 amino acid, 5 amino acid. All amino acids are bound together with the help of a peptide bond. This a long chain of amino acids. Just the strings or chains of amino acids, nothing but a protein. But we have mentioned the terms primary, secondary, tertiary, quaternary. ಇಲ್ಲಿ ಬರೀ ಚೈನ್ ಅಂತ ಹೇಳಿದ್ವಿ, ಸ್ಟ್ರಿಂಗ್ ಅಂತ ಹೇಳಿದ್ವಿ. ಆದರೆ ಆ ಪ್ರೈಮರಿ, ಸೆಕೆಂಡರಿ, ಟರ್ಷರಿ, ಕ್ವಾಟರ್ನರಿ ಅಂದರೆ ಏನು ಅನ್ನೋದ್ರ ಬಗ್ಗೆ ಇವತ್ತು ನಾವು ತಿಳ್ಕೋಬೇಕು. ರೈಟ್, ಸೊ ಸ್ಟಾರ್ಟಿಂಗ್ ವಿತ್ ದ ಫಸ್ಟ್ ಒನ್, ಪ್ರೈಮರಿ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಪ್ರೋಟೀನ್. ಓಕೆ, ಫಸ್ಟ್ ಟೈಪ್ ವಿಲ್ ರೈಟ್ ಇಟ್ ಆಸ್ primary structure of proteins. So simply here primary structure refers to when 1 amino acid, 2 amino acid, 3 amino, better to write it like this: 1 amino acid, 2 amino acid, 3 amino acid, 4th amino acid, 5th amino acid, 6th amino acid, all are bound together with the help of a peptide bond. It means the linear arrangement of the amino acids. Means we can study the positional information of the amino acids. For example, first amino acid is alanine, second amino acid is valine, third amino acid is asparagine, fourth amino acid is glutamine, fifth amino acid is glutamic acid, sixth amino acid is asparagine. Like that, in primary structure, whenever there is linear arrangement of amino acids, we can study the positional arrangement of the proteins. We can identify the first amino acid, second amino acid, third amino acid, like that in a long chain we can identify the position of every amino acid. So it is now a polypeptide. Polypeptide. Since many amino acids are joined together each other with the help of a peptide bonds. This is what we have referred in the earlier classes as a peptide bond. Peptide bond. Okay, so that is about primary structure of protein. Okay, better we write it linear arrangement of proteins, right? And it helps to know the positional information of amino acids. It means we can study the position of every amino acid. ನಾವು ಯಾವುದೇ ಅಮೈನೋ ಆಸಿಡ್ನ ಪೊಸಿಷನನ್ನು ಸ್ಟಡಿ ಮಾಡಬಹುದು. ಅಂತಹ ಸ್ಟ್ರಕ್ಚರ್ ನಾವೇನು ಕರಿತೀವಿ, ಪ್ರೈಮರಿ ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಕರಿತೀವಿ. ಓಕೆ, ಇನ್ನೊಂದು ವಿಷಯ, ಹಿಯರ್ the left end of the amino acid, this is left end, this is right end. Okay, left end, right end. Here the left end of the protein is called N-terminal. The left end of the amino acid, left end of the protein is called N-terminal, and the right end is called as C-terminal. Right end is called C-terminal. Okay, here the term N, the letter N refers to amine group, that is NH2 or NH3. Some amino acids have a NH2, some have a NH3. Here the letter C refers to carboxylic acid, carboxyl group. Means ಹೀಗೆ ಬರ್ತೀವಿ, ಅರ್ಥ ಆಗಲಿಲ್ಲ ನಮಗೆ, ಆದರೆ ಇಲ್ಲಿ ಅಮೈನೋ ಆಸಿಡ್ ಮೆನ್ಷನ್ ಮಾಡಿದ್ದೀವಿ. ಸೊ recall the side chains present in the amino acid. For example, to this carbon, at the upper side we have an hydrogen, at the lower side we have a R group, and towards the left side we have a NH2 or NH3. On the other side we have a COOH, carboxylic acid. This is one amino acid. This is one amino acid, right? So here we have amine group. This is N-terminal. We have carboxyl group. This is C-terminal, carboxyl group. Similarly, this is also having the same structure. This is also having the same structure. So all amino acids have the same structure, but just this R group is different. That's it, right? So we'll draw it for the last amino acid also. Similarly, the last amino acid has carbon, which has hydrogen as one of the functional groups, and the variable group as R2. We'll take it Rn. Since R1, R2, R3, R4, R5, R6 something, it goes to Rn. On left side we have a NH group, H2N. Here we have a COH, carboxyl group. So this is also one of the amino acid. This is one amino acid, right? Here we have amine group, that is N-terminal. We have carboxyl group, C-terminal. It means in this amino acid the carboxyl group is exposed. In this amino acid the carboxyl group is free, but towards this the amine group is bound to this amino acid. ಹೌದಾ, ಈ ಕಡೆ ಇರೋ N ಗ್ರೂಪ್ ಇಲ್ಲಿರೋ C ಗ್ರೂಪಿಗೆ ಅಟ್ಯಾಚ್ ಆಗಿದೆ. ಇಟ್ಸ್ ನಾಟ್ ಫ್ರೀ. ಕಮಿಂಗ್ ಹಿಯರ್, the carboxyl group of this amino acid, amine group of this amino acid, they are joined, they are not free. But at this side only amine group is free, unbound. Hence this end, left end is called N terminal, and the other end where the carboxyl group is free means it is not attached to any one of the functional groups, not to any one of the carbons. Hence this end is known as a C terminal. Remember, it will come for 1 mark question. Okay, 1 mark question ಕೇಳ್ತಾರೆ. What do you mean by N terminal? ಅಥವಾ left end of the protein is called N terminal. Why? Right end of the protein is called C terminal. Why? ಆ ರೀತಿ ಕ್ವಶನ್ ಬಂದಾಗ ನಾವು ಆನ್ಸರ್ ಏನು ಮಾಡಬೇಕಾಗಿರುತ್ತೆ? Just because the first amino acid has free amine group, means the amine group of the first amino acid is not bound to any of the functional groups, is not bound, it is free. Hence this end is called N terminal. Here in this case, the carboxyl group of the last amino acid is not bound to any of the functional groups, it is free. Hence the right end of the protein is called C terminal. Right? It's ನಿಮಗೆ ಗೊತ್ತಿರಲೇಬೇಕು. Right? ಒಂದ್ಸಲ better pause the video or take the screenshot of this. Take the screenshot of this and write this in your book and send it to the group. It's your homework. Okay, you should write in the same manner. It means just as a manner of practice, just for a practice. It will not come for examination. Means this diagram will not come. The questions will come. Means ಈ ರೀತಿ ಕ್ವಶನ್ ಕೇಳ್ತಾರೆ, ಆದರೆ ಈ ರೀತಿ ಡಯಾಗ್ರಾಮ್ ಹಾಕ್ಲಿಕ್ಕೆ ಕೇಳೋದಿಲ್ಲ ಅವರು. ರೈಟ್, ಡಯಾಗ್ರಾಮ್ ಹಾಕಂತ ಹೇಳೋದಿಲ್ಲ, ಆದರೆ ಈ ಕ್ವಶನ್ ಕೇಳ್ತಾರೆ. ಬಟ್ ಫಾರ್ ಯುವರ್ ಪ್ರಾಕ್ಟೀಸ್ ಐ ಆಮ್ ಟೆಲಿಂಗ್ ನೌ, pause the video, write this completely, perfectly, and click a photo and upload in the group. Right, I'll see after the class. Right, don't upload now, upload after the class. Right, okay. This is about the primary structure, where we can understand the position of every amino acid or the linear arrangement of amino acids. We can see what is the second amino acid, what is the sixth amino acid, everything we can get the clear idea of. Okay, that's all about primary structure of a protein. Shall we move on? Okay, secondary structure. Whenever we go from primary to secondary, there must be some modification, there must be some processes. Okay, so what is that process? Here, from the primary structure to form the secondary structure, there is a process called coiling. Better we'll write it first. So second one, secondary structure. Secondary structure of proteins. So here the amino acid chain forms clockwise undergoes clockwise coiling to form a helix. Mainly the helix is left-handed. Mainly the helix is means towards left hand, right to left like that. So the helix is left-handed. Artha agudilla, right? So for that I have a small model of some things So let us take some materials from the physics lab so that we can understand easily. Okay, let me go to that. So students, I'll just show you some of the things which you can see in every day, right? Better I'll write it. Amino acid, 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 right? So here whatever we can see a linear arrangement of the amino acids, that's a simple protein, or here primary structure, that is what we can see here. Imagine this is a protein. Imagine this wire is a protein. Okay, this wire is a protein that is made up of many amino acids. Imagine, okay, don't ask where is amino acid. Imagine it's an amino acid chain. Here there is no folding, there is no coiling. I just have said from primary to secondary there is a process called coiling. That the coiling is in clockwise manner. The coiling in a clockwise manner. Clockwise manner, right? So here when this primary protein starts coiling, when this primary protein starts coiling, you can better see properly. So when this primary protein starts coiling upon itself, okay, like this it will start making coils, clockwise coils. So ultimately it will form the secondary structure. Better I will show one more. This is secondary structure, where when we look at this string without coiling, it is a primary structure. It is a primary structure where we can see the position of each amino acid or the linear arrangement of the amino acids. But when it undergoes the clockwise coiling, the helical coiling, it shows the secondary structure. Okay, ನೀವು ನೋಡಿರ್ಬೋದು, ಈಗ ರೋಡ್ ಪಕ್ಕ ಮನೆ ಕಟ್ಟಿರ್ತೀರಿ, ಆದರೆ ನಿಮಗೆ ಮೇಲೆ ಕೂಡ ಮನೆ ಕಟ್ಟಬೇಕಾಗಿರ್ತದೆ. ಆದರೆ ಆ ಸ್ಟೇರ್ ಕೇಸ್ ಮಾಡಲಿಕ್ಕೆ ನಿಮ್ಮ ಹತ್ರ ಸಫಿಷಿಯೆಂಟ್ ಸ್ಪೇಸ್ ಇರೋದಿಲ್ಲ. ಆವಾಗ ಏನು ಮಾಡ್ತೀರಿ ನೀವು? ಯು ವಿಲ್ ಗೋ ಫಾರ್ ಅ ಹೆಲಿಕಲ್ ಸ್ಟೇರ್ ಕೇಸ್ ಆರ್ ರೌಂಡೆಡ್ ಸ್ಟೇರ್ ಕೇಸ್. ಏನು ಮಾಡ್ತೀರಿ? ಅಲ್ಲೊಂದು ದಪ್ಪದು ಪೋಲ್ ಇರ್ತದೆ, ರೈಟ್? ಸೊ ಯುವರ್ ಸ್ಟೇರ್ ಕೇಸ್ ವಿಲ್ ಬಿ ಲೈಕ್ ದಿಸ್. ಅಂದರೆ so like this your staircase will be. ಒಂದು ಕಂಬಕ್ಕೆ staircase ರೌಂಡ್ ಮಾಡ್ತೀರಾ, ರೈಟ್? that is how the proteins also start coiling each other. ಏನು ಇದೇ ನಿಮಗೆ ಅಮೈನೋ ಆಸಿಡ್ ಇರುತ್ತಲ್ಲ, ಅದು ಕೂಡ ಈ ರೀತಿ ಕಾಯಿಲ್ ಆಗಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ದಿಸ್ ಫಾರ್ಮ್ಸ್ ದ ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್. ಓಕೆ, ಸಿಂಪಲ್ ಅಷ್ಟೇ. When it is normal, it indicates the primary structure, that we can understand the position of every amino acid. We can see which amino acid is present at what position. But when it undergoes the clockwise coiling, that represents the secondary structure. ನೀವು ಕೇಳಬಹುದು, ಅದಕ್ಕೆ ಯಾವುದೇ ಸಪೋರ್ಟ್ ಇಲ್ಲ, ಅದು ಬಿದ್ದು ಹೋಗ್ಬೋದು, ಅಥವಾ ಈ ಕಾಯಿಲ್ ಬಿಚ್ಚಬಹುದು, ಈ ಕಾಯಿಲನ್ನು ಸಪ್ರೇಟ್ ಆಗ್ಬೋದು, ಅದಕ್ಕೆ ಯಾರು ಸ್ಟೆಬಿಲಿಟಿ ಕೊಡ್ತಾರೆ ಅಂತ ಕೇಳ್ಬೋದು. ಅಂದರೆ ಈಗ 4 ರೌಂಡ್ ಇದ್ದಾವೆ, ಹೌದಾ? ಈ 4 ರೌಂಡ್ಗಳಿಗೆ ಯಾರು ಸಪೋರ್ಟ್ ಕೊಡ್ತಾರೆ? ದ ಕ್ವಶನ್ ಅರೈಸಸ್. ಎಸ್, ಎಕ್ಸಾಕ್ಟ್ಲಿ, ಯು ಆರ್ ರೈಟ್. ಫಾರ್ ದಿಸ್ ಹೆಲಿಸಸ್, ಫ್ರಮ್ ದಿಸ್ helix to this helix, this to next, like that. Between every helix there are many hydrogen bonds. For example, here ಈಗ ನಿಮಗೆ ಒಂದು ರೌಂಡ್ ಕಾಣ್ತಾ ಇದೆ, ನೆಕ್ಸ್ಟ್ ರೌಂಡ್ ಕಾಣ್ತಾ ಇದೆ. Between these 2 rounds, what is the bond? The bond is named hydrogen bond. ಓಕೆ, ಈಗ ಈ 2 ರೌಂಡ್ಗಳ ಮಧ್ಯೆ ಇಲ್ಲಿ ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ ಇರೋದ್ರಿಂದ ಅದು ಬಾಂಡ್ ಸ್ಟ್ರಾಂಗ್ ಆಗಿರುತ್ತೆ. ಅಂದರೆ ಈ 4 ಲೇಯರ್ಗಳು ರಿಂಗ್ಗಳು ಸ್ಟ್ರಾಂಗ್ ಆಗಿ ನಿಂತ್ಕೊಳ್ತವೆ. ಮೀನ್ಸ್ ದೇ ಮೇಂಟೈನ್ ದ ಸ್ಟೆಬಿಲಿಟಿ ಆಫ್ ದಟ್ ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ದ ಪ್ರೋಟೀನ್. ಓಕೆ, ಬೆಟರ್ ವಿಲ್ ರೈಟ್ ಇಟ್. Hydrogen bonds will stabilize the secondary structure. Hydrogen bonds will stabilize the structure when there is a clockwise coiling. Here the hydrogen bonds between this and this, bond between this and this, bond between this and this, like that the bonds will keep on forming. So hydrogen bondಗಳು ಈ ರೀತಿ ಫಾರ್ಮ್ ಆಗಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗ್ತವೆ. So these hydrogen bonds make our protein more stable. Between this ring and next ring, one hydrogen bond is there, 2 hydrogen bonds are there. Between this ring and the next ring, again hydrogen bonds are there. So these hydrogen bonds will make stable the secondary structure of the protein, right? That's how the secondary structure is formed, right? One more thing we should understand. When we say secondary structure, there are 2 different types of structures formed. Okay, the next point will be here. The coiling may result in the formation of it may form alpha helices or it may form beta pleated sheets. Either it may form helix or sheets. We have a detailed explanation about this in a NEET portion. When we come for the NEET purpose, while discussing the objective questions, we will discuss this in a detail. Better I will simply tell you with the help of the same model here. Okay, so when of course it has to coil, better I will make use of our markers. So once I am done with this, I can explain better. So this is the coil here, right? So here just look at the 2 coils, 2 turns. Okay, if the bond is made between these 2, no problem, it will just make a coil. It will just make a coil like this. Better we have one more. Okay, so when bonds are made between this and this, this and this, this and this, lastly this and this, so this continues till reaching the next. So it cannot be unwound. So ಅದು ಸೆಪರೇಟ್ ಆಗೋದಿಲ್ಲ. It's completely a helix. It is called a helix, alpha helix. ಇಲ್ಲೇನಾಗುತ್ತೆ ಅಂತಂದರೆ, better we draw it better. So imagine this is the coil. Here when the bonds make between the respective coils itself, that becomes a sheet, So this is called as a beta pleated sheet. Okay, it is just for your understanding. This may not come in examination. Okay, ನಿಮಗೆ ಎಕ್ಸಾಮಲ್ಲಿ ಡ್ರಾ ಅ ಲೆವೆಲ್ ಡಯಾಗ್ರಾಮ್ ಆಫ್ ಆಲ್ಫಾ ಹೆಲಿಕ್ಸ್, ಡ್ರಾ ಅ ಲೆವೆಲ್ ಡಯಾಗ್ರಾಮ್ ಆಫ್ ಬೀಟಾ ಪ್ಲೇಟ್ಸ್, ಆ ರೀತಿ ಕ್ವಶನ್ ಕೇಳೋದಿಲ್ಲ. ಡೋಂಟ್ ವರಿ ಅಬೌಟ್ ದಿಸ್, ಜಸ್ಟ್ ಟು ಮೆನ್ಷನ್ ದಿಸ್, ಇಟ್ಸ್ ಎನಫ್. ಓಕೆ, ಅದು ನೀಟಿಗೆ ಬಂದಾಗ ಅಲ್ಲಿ ಡಯಾಗ್ರಾಮ್ ಬರೋದಿಲ್ಲ ಅಫ್ಕೋರ್ಸ್, ಅಲ್ಲಿ ಬೇರೆ ನಾವು ಅದ್ರ ಮೇಲೆ ಕ್ವಶನ್ ಕೇಳ್ತೇವೆ, ಯಾವ ಬಾಂಡ್ ಅಂತ ಕೇಳ್ತೀವಿ, ಅಲ್ಲಿ ನಿಮಗೆ ಈ ಹೈಡ್ರೋಜನ್ ಬಾಂಡ್ ಗೊತ್ತಿದ್ದರೆ ಸಾಕು, ದಟ್ಸ್ ಇನಫ್. ಓಕೆ, ದಿಸ್ ಜಸ್ಟ್ ಫಾರ್ ಯರ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್. ಓಕೆ, ಸೊ ಲೈಕ್ ದಿಸ್ ದ ಕಾಯಿಲಿಂಗ್ ಮೇ ಫಾರ್ಮ್ alpha helix, the coiling may form beta pleated sheets, right? That is the brief idea about a secondary structure. Okay, next, after this coiling another level of coiling occurs that leads to the formation of tertiary structure. Tertiary structure of proteins. Okay, here coiling and superimposition of alpha helix or beta pleated sheets. Beta pleated sheets result in a ball like structure which is referred as tertiary structure. ಸಿಂಪ್ಲಿ ಏನಂತಂದರೆ, ಈಗ ಮೊದಲು ನಾವೇನಿಲ್ಲಿ ಅಲ್ಪ ಹೆಲಿಕ್ಸ್ ಅಥವಾ ಬೀಟಾ ಶೀಟ್ಸ್ ಆಗಿರ್ತವೆ, ಆ ಶೀಟ್ಸ್ಗಳು ಏನಾಗ್ತವೆ ಅಂತಂದರೆ ದೇ ವಿಲ್ ಮೇಕ್ ಅನದರ್ ಲೆವೆಲ್ ಆಫ್ ಕಾಯಿಲಿಂಗ್. ಅದು ಒಂದು ಬಾಲ್ ಥರ ಆಗುತ್ತೆ, ಮೊದಲು ನಿಮಗೇನಿತ್ತಂದರೆ ಪ್ರೈಮರಿ ಸ್ಟ್ರಕ್ಚರಲ್ಲಿ ಈ ರೀತಿ ಒಂದು ಥ್ರೆಡ್ ಇತ್ತು, ಲೀನಿಯರ್ ಚೈನ್. ಅದೇ ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್ ಏನಾಗುತ್ತೆ, ಕಾಯಿಲ್ ಆಗುತ್ತೆ, ಐದರ್ ಶೀಟ್ ಆಗುತ್ತೆ ಇನ್ ದ ಸೇಮ್ ಮ್ಯಾನರ್. ಬಟ್ ಇನ್ ಟರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್, the secondary protein will undergo coiling, superimposition. Imposition ಅಂದರೆ upon another. ಓಕೆ, ಸೂಪರ್ ಇಂಪೋಸಿಷನ್ ಒಂದ್ರ ಮೇಲೆ ಒಂದು ಬೀಳೋದು ಅಂತ ಹೇಳ್ತೀವಿ ನಾವು. ಓಕೆ, ಅಂದ್ರೇನು, ಆ ಒಂದು ಲೇಯರ್ ಇನ್ನೊಂದು ಲೇಯರ್ ಮೇಲೆ ಬೀಳುತ್ತೆ. ಸೊ ದಟ್ ವಿಲ್ ಮೇಕ್ ಅ ಬಾಲ್, ಅದೊಂದು ಉಂಡೆ ಥರ ಕಾಣುತ್ತೆ. ದಟ್ ವಿಲ್ ಲೀಡ್ ಟು ದ ಟರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್. One more thing you should remember, this is required for most of biological activities of proteins. ಅಂದರೆ ಒಂದು ಪ್ರೋಟೀನ್ ಕೆಲಸ ಮಾಡಬೇಕು ಅಂತಂದರೆ ಆ ಪ್ರೋಟೀನು ಆ ಟರ್ಷರಿ ಫಾರ್ಮಲ್ಲಿದ್ದರೆ ಮಾತ್ರ ಅದು ಮೋರ್ ಆ್ಯಕ್ಟಿವ್ ಆಗಿರುತ್ತೆ. ಪ್ರೈಮರಿ ಸ್ಟ್ರಕ್ಚರ್ನಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡಬಹುದು, ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್ನಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡಬಹುದು ಬರೀ. ಆದರೆ ಆ ಪ್ರೋಟೀನ್ ಕೆಲಸ ಮಾಡಬೇಕು ಅಂತಂದರೆ ಅದು ಯಾವ ಫಾರ್ಮಲ್ಲಿರುತ್ತೆ ಅಂತಂದರೆ ಐದರ್ ಪ್ಲಾಂಟ್ ಆರ್ ಅನಿಮಲ್, ದಟ್ ವಿಲ್ ಬಿ ಇನ್ ದ ಫಾರ್ಮ್ ಆಫ್ ಅ ಟರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್, ರೈಟ್? ಜಸ್ಟ್ ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್ ಹೋಗಿ ಏನಾಗುತ್ತೆ, ಅದು ಸೂಪರ್ ಕಾಯಿಲ್ ಆಗುತ್ತೆ. ಸೂಪರ್ ಕಾಯಿಲ್ ಆಗಿ ಅದು ನೆಕ್ಸ್ಟ್ ಒಂದು ಕಾಯಿಲ್ ಮಾಡುತ್ತೆ. ದಟ್ ಕಾಯಿಲ್ ಲೀಡ್ಸ್ ಟು ದ tertiary structure. Okay, this is about a tertiary structure. Lastly, the last type of protein depending on the structure, the fourth one, quaternary structure. Quaternary structure of proteins. So when we have seen from primary structure, secondary structure, tertiary structure, there were levels of coiling has increased. Means we observed different levels of coiling. But here coiling doesn't matter. The coiling must happen. But to consider this quaternary, of all the proteins are, they must be in a ಟರ್ಷರಿ ಟು ಫಂಕ್ಷನ್, ಈ ರೂಪದಲ್ಲಿ ಇರಲೇಬೇಕು. ಆದರೆ ನಾವು ಕ್ವಾಟರ್ನರಿ ಅಂತ ಹೇಳಬೇಕಾದ್ರೆ ಅಲ್ಲಿ ಒಂದಕ್ಕಿಂತ ಜಾಸ್ತಿ ಪ್ರೋಟೀನ್ಗಳು ಇರಬೇಕು. ಹಿಯರ್ ಇಟ್ ಈಸ್ assemblage of, it is the assemblage of more than polypeptides. Remember, more than one polypeptide properly. ಓಕೆ, ಯಾಕಂತಂದರೆ ಪ್ರೈಮರಿ ಸ್ಟ್ರಕ್ಚರಲ್ಲಿ ನೋಡಿದಾಗ ಅಲ್ಲಿ ಒಂದೇ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಇತ್ತು ವಿತ್ ಸಮ್ ಅಮೈನೋ ಆ್ಯಸಿಡ್ಸ್, ರೈಟ್. ಸೆಕೆಂಡರಿ ಸ್ಟ್ರಕ್ಚರ್ ನೋಡಿದಾಗ ಅಲ್ಲಿ ಕೂಡ ಒಂದೇ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೇನ್ ಇತ್ತು, ಹೌದಾ? ಒಂದೇ ಇದೆ, ಅಲ್ಲೂ ಒಂದೇ ಇದೆ, ಕಾಯಿಲ್ ಆಗಿರ್ಬೋದು ಆದರೆ ಒಂದೇ ಇದೆ ವಿತ್ ಸಮ್ ಅಮೈನೋ ಆ್ಯಸಿಡ್ಸ್ ಆನ್ ಇಟ್, ನಾಟ್ ಸಮ್ ಮೆನಿ ಅಮೈನೋ ಆ್ಯಸಿಡ್ಸ್ ಆನ್ ಇಟ್, ಹೌದಾ? When we see the tertiary structure also, we have seen the same strand, a single strand, ಒಂದೇ ಸ್ಟ್ಯಾಂಡ್ ಆಗಿತ್ತು with a few more amino acids, right? ಅಂದರೆ one chain in primary structure, the same single chain in secondary structure, but it is called the same single chain in tertiary structure with a more coiling. But in quaternary, the polypeptides must be more than one. Then only we can consider that it is a quaternary structure. For example, I have shown one primary structure and the secondary structure here. Primary, secondary. When it makes a ball of the amino acids, polypeptide chain, that we consider as a tertiary, right? But when more than one polypeptides join each other, that we consider as a quaternary structure. So this is one tertiary structure, this is one tertiary structure, ಒಂದು ಎರಡು ಕೂಡಿಸಿದ್ರೆ it becomes quaternary structure. Hope it's clear now, right? ಅಂದರೆ ನಮ್ಮ ಹತ್ರ ಪ್ರೈಮರಿ ಇದ್ದಾಗ ಇಲ್ಲಿ ಬರೀ ನಾವು ಸಿಂಗಲ್ ಅಂತ ಹೇಳ್ಕೊಂಡ್ವಿ, ಸೆಕೆಂಡ್ರಿ ಅಂದಾಗ ಕಾಯಿಲ್ ಅಂತ ಹೇಳಿದ್ವಿ, ಟರ್ಷರಿ ಅಂದಾಗ ಅದನ್ನು ಒಂದು ಬಾಲ್ ಮಾಡಿದ್ವಿ, ಕ್ವಾಟರ್ನರಿ ಬಂದಾಗ ದೇರ್ ಮಸ್ಟ್ ಬಿ one ball of one amino acid, one coil, another ball of another amino acid, right? So when two comes each other, it is a quaternary structure. ಅರ್ಥ ಆಯ್ತಾ? ಸಿಂಪಲಿ ಬರೀ ನಮ್ಮ ಹತ್ರ ಚೈನ್ ಇರಬೇಕಾದ್ರೆ, ಅಂದರೆ ಯಾವುದೇ ಕಾಯಿಲ್ ಆಗಿಲ್ಲ ಏನೂ ಆಗಿಲ್ಲ, ಅಲ್ಲಿ ಲೀನಿಯರ್ ಚೈನ್ ಇರಬೇಕಾದ್ರೆ ಅದಕ್ಕೆ ನಾವು ಹೇಳ್ತೀವಿ ಪ್ರೈಮರಿ ಪ್ರೋಟೀನ್ ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಅದು ಯಾವಾಗ ಕಾಯಿಲ್ ಆಗ್ತದೆ ಇನ್ ಅ ಕ್ಲಾಕ್ ವೈಸ್ ಮ್ಯಾನರ್, ಅದಕ್ಕೆ ನಾವು ಸೆಕೆಂಡ್ರಿ ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಆ ಕಾಯಿಲ್ ಮತ್ತೆ ಕಾಯಿಲ್ ಆಗಿ ಯಾವಾಗ ಅದೊಂದು ಬಾಲ್ ಥರ ಕಾಣುತ್ತೆ, ನಮಗೆ ಒಂದು ಬಾಲ್ ಥರ ಕಾಣುತ್ತೆ, ಅದಕ್ಕೆ ನಾವು ಟರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್ ಅಂತ ಹೇಳಿದ್ವಿ. ಆದರೆ ಆ ಕ್ವಾಟರ್ನರಿ ಸ್ಟ್ರಕ್ಚರ್ ಆಗಬೇಕಾದ್ರೆ ಈ ಸುಮಾರು ಒಂದಕ್ಕಿಂತ ಜಾಸ್ತಿ ಟರ್ಷರಿ ಸ್ಟ್ರಕ್ಚರ್ಗಳು ಕೂಡಿಕೊಂಡು ಅದೇನೋ ಒಂದು ಕಾಂಪ್ಲೆಕ್ಸ್ ಬಾಲ್ ಆಗುತ್ತೆ. ದಟ್ ವಿ ಕ್ಯಾನ್ ಕನ್ಸಿಡರ್ ಇಟ್ ಆ್ಯಸ್ ಅ ಕ್ವಾಟರ್ನರಿ ಪ್ರೋಟೀನ್. ಕ್ವಾಟರ್ನರಿ ಪ್ರೋಟೀನ್. ಓಕೆ, ಸೊ ದಟ್ ಇಸ್ ಹೌ ವಿ ಕ್ಯಾನ್ ಸ್ಟಡಿ ದ ಡಿಫರೆಂಟ್ levels of coiling, different types of proteins based on the structure, arrangement of the coiling, right? So better once pause the video, take screenshot, that becomes your notes. Other than that there is nothing. One more thing we should understand here. For example, here all of you are aware of our hemoglobin. Better I'll write with other pen to make it clear. Here we have shown two colors. It means two polypeptides. Better I will take two more. One here and another here. I can see 4 polypeptides. It means ಈ ರೀತಿ ಇತ್ತು, ಆಮೇಲೆ ಈ ರೀತಿ ಆಯಿತು, ಆಮೇಲೆ ಈ ರೀತಿ ಆಯಿತು, ಆಮೇಲೆ ಈ ರೀತಿ ಆಯಿತು, ರೈಟ್? primary, secondary, tertiary, quaternary. Okay, the hemoglobin measure content of our blood contains 4 polypeptides. 4 polypeptides of 2 types. Andre 2 of alpha type, 2 of beta type. ನೀವು ಹೇಳ್ತೀರಿ ಮನೆಯಲ್ಲಿ 4 ಜನ, ನಮ್ಮ ಅಪ್ಪನಿಗೆ 4 ಜನ ಮಕ್ಕಳು ಅಂತ ಹೇಳ್ತೀರಿ. ಅದರಲ್ಲಿ 2 ಗಂಡು ಮಕ್ಕಳು 2 ಹೆಣ್ಮಕ್ಳು ಅಂತ ಹೇಳ್ತೀರಿ. 2 are male, 2 are female. Like that, in hemoglobin there are 4 polypeptides of 2 types. 2 are of alpha type, alpha subunits or alpha polypeptides, and 2 are of a beta type or beta polypeptides. So altogether they will make a hemoglobin. 4 polypeptides, 4 are of 2 types, 2 alpha type and 2 beta type. So they will make a hemoglobin of 4 polypeptides, right? Similarly, another example, human insulin. That's also a protein, remember. Hemoglobin is a protein, insulin is a protein. Insulin is made up of 2 polypeptides. Insulin is made up of 2 polypeptides, 1 polypeptide, 2 polypeptides, right? And the 2 are joined each other by disulfide bonds. This we'll study in second PU biotechnology. I'm just giving for example, okay, not for study. This is there in our first PUC. Remember this example compulsorily. Whenever this question comes in examination, we have to write this example. Don't write this, not need. You can remember this for need purpose, but this will come in second PUC. Okay, but for now, again, this may come in human physiology, discussing digestion and all other things, but that may come in human physiology. But for this chapter, for mainly for the quaternary structure, this example is mandatory, compulsory. Idu nenapiralebeku. Okay, hemoglobin is made of 4 polypeptides. Insulin is made up of 2 polypeptides. Made of 2 polypeptides means 2 chains. Okay, hope the concept is clear of the structure of a protein, right? In the next class, we will discuss, we'll take another biomolecule. With this we have finished the proteins. Proteins are completed. Another details like types of proteins based on the chemical nature we will discuss during NEET discussion. When we come to NEET classes after completing the theory portion, we will discuss other concept about protein in NEET portion, NEET classes. But for as of theory classes, in next class we will take up another biomolecule. Next to proteins, we will take up carbohydrate. Okay, so glucose, carbohydrates, sugars, sugarcane juice, whatever we have, sugar in your home, tea, we will discuss all that in a next class. Okay, ಇನ್ನೂ ಏನು ಅರ್ಥ ಆಗಿಲ್ಲ ಅಂತಂದರೆ ಆದಷ್ಟು ಬೇಗ ಇವತ್ತೇ ಗ್ರೂಪಲ್ಲಿ ಡೌಟ್ಸ್ ಕೇಳಿ ಡೌಟ್ಸನ್ನು ಕ್ಲಿಯರ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಓಕೆ, ಇವತ್ತಿನ ವಿಡಿಯೋ ನಾಳೆಗೆ ನೋಡಿ, ಡೌಟ್ ನಾಳೆ ಕೇಳಿಬಿಟ್ಟು ಆಮೇಲೆ ಮತ್ತೆ ವಿಡಿಯೋ ಪ್ರೈವೇಟ್ ಆಯಿತು ಅಂತ ಕಂಪ್ಲೇಂಟ್ ಮಾಡಬೇಡ್ರಿ. ಆದಷ್ಟು ಬೇಗ ಇವತ್ತಿನ ವಿಡಿಯೋನ ಇವತ್ತೇ ನೋಡಿ ಆ ಡೌಟ್ಸನ್ನು ಇವತ್ತೇ ಕೇಳಿ ಮುಗಿಸ್ಕೊಂಬಿಡಿ. ಓಕೆ, ಸೊ ಬೆಟರ್ ಪುಟ್ ದ doubts in your group so that we are able to come back to you with a solution, right? So we'll see you in the next class. Till then, keep learning. Once again, thank you. Bye bye.